Alrighty, thank you everyone for coming. We're here in HLS215 to really talk about how Merck is using sort of state-of-the-art biological foundation models to improve drug discovery. My name is Aaron Friedman. I'm a principal product manager here at AWS in our Health AI organization. And I'm joined by Matt and Danny, who you will hear from shortly. This is a talk that's near and dear to my heart. I'm a computational chemist by training years ago in my doctorate. And 2 years ago, we launched AWS HealthOmics, which was a service that originally we were really focused on omics and bioinformatics work. But what was really interesting at the time is there were the proliferation, and we'll talk about this in a sec, of bio-FMs. And so what we found over the last couple of years is not only does what we were building for the bioinformatics community in resonating there, it also had broad applicability in this space. But at the end of the day, we hear a lot of talk about architecture, and I thought Matt gave a great overview this morning on those architecture building blocks, things like compute and storage and databases and inference now. And those aren't going to go away. But at the end of the day, architecture and the things that we're building with are actually means to solve business problems, business objectives. And in our space, it's really about what are the scientific questions that our customers are trying to solve, right? Things like help me design novel antibodies to target specific diseases of interest. What new mechanisms of actions come from these mutations? And I have a large compound library, and I want to screen my compounds on predictive protein structures and use that information to better inform lead optimization or hit to lead really throughout the discovery pipeline, with the end goal, right, of discovering new therapeutics and improving human health. And what's really exciting to me is being able to get to work with so many of these companies and organizations that are mission-driven and have such a tangible impact on how we live our lives, how our loved ones live our lives. It's not lost on me that the work that these organizations do impacts all of us, and it's just really exciting to be working with everyone. So drug discovery in general, right? Pharmaceutical organizations, they've existed for long periods of time, right? We've had, penicillin and aspirin and many drugs for a while. But I think what's really exciting is over the last couple of decades, you've seen an emergence of computation as a way to augment discovery. This has resulted in 2 Nobel Prizes in the last, say, decade or so. First on more physics-based simulations in 2013 with Martin Karplus and that team. And then more recently this year, really on protein prediction and protein generation with both AlphaFold 2 and all of the work that David Baker's group has done up at the University of Washington. And what this gives us the ability to do is really to augment what has been done for decades. We can get more data points faster, and we can use those data points that we generate in the lab to better inform what we do subsequently. It allows us to break through bottlenecks. It allows us to, really sample and test hypotheses that we, may not have been able to do before if we were following a more serial approach in the lab. And so the power of computation in this space is something I think will continue to emerge. And of course, right, we talk a lot right now about generative AI as it pertains to all areas of life. But it has a really nice impact here in life sciences research. And if you go back to those scientific questions that we talked about a couple slides ago, there are AI-based applications that should be able to improve how we answer all of those questions in areas like drug design, biomarker discovery, target assessment, and so on and so forth. And so within these groups, right, you have a bunch of different types of algorithms and models emerging, from RNA and DNA-based algorithms all the way to protein models, but also ways of assessing, specific properties like immunogenicity, helping design novel antibodies, and then going all the way through and thinking about things even beyond discovery in the clinic, things like digital twins, et cetera. There's a lot of different, types of models and a lot of amazing work that is going on really across the spectrum to accelerate drug discovery. And if we zero in, right, and this slide is already out of date because models are being released every week, right? In the last month, we've had Boltz 1, we've had, Chi 1R come out, just a ton of new models that continue to emerge. But if we look in sort of the protein design and discovery space, there's already a whole set, right? And as an organization, you want to be able to test these models. You want to be able to run them at scale. You want to be able to track the lineage, right? Many models, many versions. I want to swap things in and out, but I also need to sort of know what I did before and how those things relate so I can make assessments of, what, types of scientific recipes are giving me the best value. And so as we continue to, advance in this industry, orchestration becomes a really interesting challenge. And we heard, Matt talk today about inference as this fourth building block, and I think this is really important, right? We're doing a lot of model training right now. These models, right, continue to emerge. We're doing a lot of fine-tuning as well in this space, right, as part of, say, a design, build, test, learn cycle. But if you think about what we really need to do at scale in this space, it's to make many, many predictions and use that information to inform the next steps that we can take in the lab, the next best action. And so there are really two types of inference, right? There's sort of this interactive inference. I'm testing things out. Maybe I've spun up a SageMaker notebook or an endpoint, and I'm hitting, models with different parameters and really trying to optimize. And so you're looking for this really fast, iterative feedback experience. But then when you're ready, there's this whole batch approach, right? Now I need to screen 1 million compounds with DiffDock, or I need to generate, 500,000 new antibody or nanobody scaffold or potential structures that bind to a target antigen. And so that's where batch inference comes into play, right? You have multiple models, you have multiple steps, you need to do this at scale. And I think one thing that's also really important here is you want to do this efficiently. You can leave up endpoints, you can leave things up all the time, but what you get is a very low resource utilization, so the actual price performance value that you get out of these tools is much less. But when you run these in batch mode, which is more of a traditional high performance computing approach, you can get very high resource utilization and really, scale up very quickly and just as importantly, scale down. And so now, if we think about when we talk to customers, this is a lot of what they told us. And, okay, we want to use these tools. They help us provide novel data points. But how do we get started? How do we scale? And so we've really bundled this into sort of 4 major areas. First is, how do I quickly get started? What are those scientific recipes that have been proven either, in my own group, maybe proprietary types of tools and uses. Also the open source community. There's a vibrant open source community in this space. But, there are certain things that I want to do, in a specific way. Maybe I want to use a different nanobody scaffold or an antibody scaffold in RF diffusion, for example. So I need to be able to adapt these recipes, and maybe I want to add a couple property predictions at the end or scoring functions that can, add physics-based calculations to these, multimeric predictors that you can get with, say, AlphaFold 2. And then once I figured all that out, I want to scale this up. And then I want to bridge across data silos. I want to be able to share this data with collaborators. And so how do we do all these things? And if you think about the normal way of doing it, or what you would be doing today, it's a lot of architecture. You have to go, and I was an SA in my past life, you'd have to go, right, build your endpoints, you'd have to manage all of your permissions, you'd have to write your own code, your own orchestrator, you'd have to do all these different things. And so at the end of the day, you have a very robust solution that you can build on AWS, of course. That's a lot of time that organizations were spending doing things that weren't actually differentiating their business, which is the actual science. And so this, I'd say about 3 years ago at this time, was why we built HealthOmics. And I think this is actually the only architecture slide in the entire talk, and that's purposeful, because what we want to do in a lot in these cases is get our customers and end users away from having to really worry about a lot of these things. And so what we want to get to is inference effectively with one API call. And if you think about historically what you would have to do, right, you'd containerize your models, you get them up there, you then write some orchestration script, and it can be in a domain-specific language like Nextflow, which a lot of the bioinformatics and just general life sciences communities gravitating towards, or something like MLflow, or maybe you're using step functions. But you have your tools, your containers, and then you need to run the things. And so you would have to manage your shared file systems. You'd have to manage the orchestration, the failures, the retries, et cetera. And you'd have to do all these things. And really, at the end of the day, though, all you want to do is say, here are the tools that I have. Here's the data that I have. Here's the recipe I want to run. Please just run it. And so more and more, what we want to do is distill this down into a single API call. And the way that we work with customers on this is really in two approaches. The first one, which is the majority of what our customers do and what Mark will be talking about a little bit, is these private workflows where you can build your own, you can define your own scientific recipe. But for those who want to just get started, we have ways of doing this as well for, say using AlphaFold to just, default predictions, right? Give me your sequence, you get an output a little while later, nothing else for you to do. But if you want to configure it, a lot of our customers end up in the private workflow space. So back to where we were talking about, what we found is not just for bioinformatics, HealthOmics has a huge value that we found from customers in the specific space of combining high performance computing with at-scale inference for the purpose of orchestrating a lot of these different types of protein language models and broader foundation models and just general AI models. So let's look through these 4 pieces. First, right, starting with a proven recipe. Now, I mentioned ready-to-run workflows, right? That's something that you can use. We have the ability to do that. But our team has actually made a lot of investments in a set of open source tools and repositories. It's the drug discovery workflows repository on AWS samples that has things like AlphaFold and Amplify and RF diffusion and protein MPNN. So you can start very easily and actually see the recipe as it sits and know exactly what it's going to do. And with this, we have a one-click deploy script as well that can immediately get these into your AWS accounts. So you've started the recipe. Well, now I want to adapt it to my needs. So what do? You go into the code if you need to, right? This is more on the low level side, but there's no infrastructure you're changing here. This is literally just defining your business logic, in this case in Nextflow. And so what you can do is you can basically say, oh, okay, I have my set of steps on the left. I have maybe my default config files on the right. Maybe I want to change the random seed. Maybe I want to add relaxation to my AlphaFold predictions. You can do all this really easily, right? You've gone in your code, one push again. It's now in your registered as a workflow within HealthOmics, and you can just run it. And then you've defined it, you've done it, and so the next thing you do is maybe you want to share this data with your collaborators. And this is something that over time we've realized has increased importance, right? Particularly between organization collaborations, but also within organizations, right? If you're a pharma and you develop something somewhere, you don't want another person to go develop the same thing. That's just wasted resources. You want to just be able to create a catalog and share that catalog with others. And so we have things here like workflow sharing and being able to collaborate. If, for example, you're working between organizations in a collaboration and you want to run a scientific recipe that, say, a collaborator, maybe a tech bio startup, has developed on your own data, but you don't want to share that data, you can do that because they can share the workflows directly with you, and you can just run the data in your own account. And the collaboration, whoever you're partnering with doesn't ever have to see it. So things like being a secure collaboration, acting as effectively an honest broker, in this case, again, can be really powerful from fostering collaboration and enabling you to do your science in the way that you need to. And then, I kind of just already talked about it. This is how easy it is, right? You just go, you say, hey, I want to share this with this, this group, this AWS account, and the access is, once they accept it, it's granted, you can revoke it, you have full control over these things. But collaboration is super important. And then once you've done all this, you need to scale, right? If we go back to batch inference and how do we actually run these analyses at scale, there's this idea that I like to call serverless science, right? We hear a lot about serverless as one of the key things is being able to scale to zero and scale to whatever you need. We can do that here too. You can run thousands of compute predictions simultaneously, and then when you're done, you don't want to pay anything. And so this, again, gets really powerful as you start to think about orchestrating more and more of these complex scientific recipes, things like, say, going from an RF diffusion to protein MPNN to AlphaFold for the purposes of, say, protein engineering and design. And so what I'm really excited about is all this stuff, right? You actually don't have to go build, right? You can just use as a managed service, and it's really API driven. And with that, I think now I'd like to transition it over to Merck, because what they're going to talk about are business outcomes and scientific outcomes, not necessarily the architecture. I think that's really, again, what is exciting about all this. So I'd like to hand it off to Matt Studney, VP of R&D IT at Merck.
Thanks. Thanks, Aaron. It's great to be here, everybody. So again, my name is Matt Studney. I lead information technology for Merck Research Laboratories. And what that means is I have responsibility to deliver all of the capabilities for our scientists in discovery, developmental sciences, clinical research, pharmacovigilance, regulatory, business development, medical affairs. We structure our teams into product and capability models that are broken up into 3 value teams, 20 product lines, 70 products, and 1,000 plus applications. And we've been partnering with Amazon Web Services for more than a decade. And when we started partnering with them, we were basically going after the reality that we want to be a biopharmaceutical company that develops, discovers and develops drugs, not necessarily in the business of managing infrastructure. So it was a heavy infrastructure play, really good partnership. But over time, we started to shift that to more meaningful capabilities and partnerships. We've been in the process of refactoring our applications, lifting and shifting them. In research alone, we've done that with over 400 of those 1,000 applications. We've just moved over 50% of our high-performance compute capabilities from our on-prem servers to AWS. And what that means is our data is going with it. We're a company that's over 100 years old. So now we've got all this data sitting on AWS. We have almost unlimited compute. We have high-performance compute capabilities. And now they've enabled capabilities like the omics platform, and we've been working with them to unlock insights that we could never do before. And so we're heavily investing in capabilities to improve what targets we go after, how we build that composition of matter, making sure that it's as safe as possible with billions of data sets. And Danny Bitton on my team has been working closely with Aaron and his team to enable some of those workflows. And really what we want to do is make sure that this is seamless from data generation to the user experience of the scientists to come up with those scientific outcomes. And as the world's been changing the last 3 years, AWS has been moving really from that infrastructure partner to a capability building partner to unlock ideas and concepts and outcomes and insights that we've never been able to do before. So I'm really excited to bring Danny to the stage, who's going to walk you through a couple of those workflows that have been really powerful and really successful. So thank you.
Thank you, Matt. Good afternoon, everybody. So in this part of the session, I will try to focus on the scientific outcomes and how HealthOmics services actually helped us to accelerate protein design with So I will start with the overview on protein design and how AI and machine learning methods are used for design and optimization, and how we partnered with the HealthOmics team to drive these workflows. So broadly speaking, the HealthOmics team enable us to streamline and democratize workflows, either ready to run or bespoke, but also to scale our compute and shorten the compute time, but also to increase the size of inference, which means that we can do more computation that we couldn't do before. So I will again focus on the science. So as you know, we are mostly made of water and proteins. And we have about 35 trillion cells in our body, and each cell is actually a protein factory. So proteins are everywhere, and they really play critical cellular functions and mediate all the key processes in the cell. They are the structural components that build our tissues. They are the hormones and the transport and the signaling molecules, as well as the immune system molecules that are actually defend us from bacteria and viruses. They are also the enzymes that catalyze all the reaction in our body. So therefore, if there is any issue with the protein sequence or structure or the function, then it's likely to lead to a disease. So we, in a pharmaceutical company and any other pharmaceutical company, what we do is design drugs to target these proteins. So when it comes to foreign proteins, they can be used as vaccines to prime our immune system and protect us from future infections. So we can design small molecules to bind to these proteins, but we can also design proteins to target these proteins. So we can conclude and say that proteins are the primary drug and vaccine targets and can also be used as drugs. And here I listed a few examples. We have the pembrolizumab as an antibody. It's a protein that binds to a receptor that sits on the immune cell, and basically that's how we get the therapeutic effect by interfering the binding between the immune system and the cancer cells, and basically exposing the cancer cells to elimination by the immune system. Similarly, we have the PCSK9 enzyme that is implicated in the metabolism of cholesterol, and we can design a cyclic peptide inhibitor, which is again a protein that interferes with the binding with the cholesterol receptor and basically reduce the cholesterol levels in the blood. When it comes to viruses like HPV, the coat proteins can actually be used as vaccine. And what you can see is that following vaccination, the entire virus is basically covered by antibodies. So I did mention that enzymes catalyze all the reactions in our body. In fact, enzymes are chemical factories. We can exploit that and basically use enzymes and re-engineer them to work in industrial conditions, different temperature, different pH, and basically use them to manufacture drugs. So given all these diverse functions of proteins, it begs the question why we need to design and optimize in the first place. And the answer is quite straightforward. Basically, the attributes of natural proteins are very different than the properties that we want drugs to have in terms of stability, solubility, and ease of manufacturing. Also, there are a lot of proteins out there in the wild, in nature, but we often want to create new proteins with new desirable functions and properties. And if we were to do that in the lab, it would be tedious and expensive. So what we want is to create these computational pipelines to create thousands of designs on the computer and basically reduce the number of designs that we actually take to the lab. So I did mention, and also Aaron, that recent years have seen a dramatic increase in AI and machine learning methods that completely revolutionized the field. And most notably are the deep learning methods like Google DeepMind AlphaFold or the Rosetta Fold from the David Baker's lab or the MetaFold the ESM Fold from the Meta group. But all they do is one thing. They are able to basically predict a three-dimensional structure of a protein directly from its sequence. The Baker's lab also developed other protein language models that enable to do the other way around. Basically go from a three-dimensional structure into a sequence. They also developed diffusion models that enable to generate completely new designs that don't exist in nature. And it's very similar to actually the OpenAI DALL-E tool that allows you to create AI images directly from text prompts. So here you can basically use protein prompts, yes, and create new structures. So we teamed up with Aaron's team a while ago to combine basically all these methods into a pipeline, a generative AI pipeline that enabled to do thousands of designs at scale by a click of a button. So our scientists can basically input a structure of interest or a prediction, and basically they can specify the contig or the area of the protein that they want to retain, and then we can create designs around that. So this can be used for creating new vaccines, enzymes, peptides, as well as different protein binders for therapeutic or for diagnostic. So when we say protein language model, it's very similar to a large language model. The only difference is actually the input. So in a language model, the input is a large corpus of text, and we try to predict a word from the context. With protein language model, the input is actually a large corpus of sequences, and we try to predict the relevant amino acid from the context. When it comes to generative AI, if we use ChatGPT, we can generate new text, new content. And here, in the protein space, we can use GPT-like models. For example, Progen 2, which is a protein language model from Salesforce. And we can basically fine-tune it in order to basically generate new designs. The way that it works, we have a data preparation aspect where we tokenize certain sequences that we care about, and then we fine-tune the model to predict the next token. And then what the users can do is basically use the token as a prompt and then generate new designs. Right, we can also use protein language models for antibody discovery. So The traditional approach to discover antibodies is basically to take a target protein, inject it to mice or rabbits, and then do a lot of binding assays and experiments in the lab, and then end up with a mouse antibody that binds to your target. Now, this is not a drug yet. We need to humanize it. And the way to humanize it today is to take the relevant parts from the antibody and basically cut and paste it into a human framework. Now, there are two problems with this approach. One is that only 1 or 2 people, in our company at least, know how to do that. And second is that you do a lot of experiments way before you get into a single drug candidate. So to address that, we developed an antibody language model, a whole actually platform for guiding antibody engineering decision that basically allows to do humanization at scale using the first antibody language model in the world, and that we also published, and also some other regression models. So we can do a lot of designs at scale way before we actually go to the lab. And the way to do that is simply we took a database, a public database with 40 million human sequences, and then the user can simply input 1 or more antibody sequences, non-human one for mouse or rabbit, and then it will end up with humanized lead candidates. And of course, in order to democratize the approach, as I mentioned, only 1 or 2 people in the company know how to do that. Now we can basically do that again through a very intuitive user interface. Now, when it comes to target design or enzyme diversification, we created a new platform called Autobahn that basically allows to do collaborative protein annotation and design. And it's similar to a computer-aided design tool for proteins. So basically, it integrates multiple internal and external sources, including protein language models, including AlphaFold. And one use case is that basically when we have a target protein, we can't really express it as is, and we usually need to design it. So we add all kind of tags to it and a linker to link them together. And that process basically allows to purify the protein later. Now, following purification, we need actually to cut that linker in the middle, shown in orange. And if that process is not efficient, it may lead to low yield of proteins, and then we don't have enough proteins to do assays on. So one way to do that is to do a lot of sequences, to design a lot of sequences, and then look manually on AlphaFold predictions and try to really find the best linker that we can have, the most stable one. But that's where we, again, teamed with the HealthOmics team, and basically to do molecular dynamics. Now, molecular dynamics is a virtual experiment where we basically simulate how the atoms interact with molecules over time. So we do a lot of simulations in microseconds, and we can use that basically to model the interactions between a protein and a drug, or to optimize that linker that I just showed, or to identify completely new cryptic pockets that we can design drugs to. Now, this is very computationally expensive, and we will not be able to do that before without the help of HealthOmics. And it looks something like that. You have the different linkers, basically, and you evaluate the stability of that linker, and then you choose the best linker, and then you get the highest yield of proteins from your experiment. So I did mention biocatalysis in the beginning, where we can take an enzyme, we can re-engineer it to work in more stable conditions, in industrial conditions. And biocatalysis is a great way to basically do green chemistry, more environmental friendly, but it's also reducing the number of steps to manufacture a drug and therefore reduce the manufacturing costs. The basic principles of biocatalysis are very simple. We start with an enzyme sequence, and then we basically introduce a lot of mutations. So we create a library of enzymes that we express in bacteria, and then we actually make that enzyme and we test its activity. Now, we can inject machine learning models to improve the choice of mutations, but we can also use molecular dynamics, as I mentioned, to model essentially the interactions between the substrate and the enzyme. And we can do that in Autobahn, where we basically have a lot of information from the protein language models and multiple sequence alignment, but also regression models that allow us to identify which mutation is likely to be beneficial and improve the enzyme activity. And for the last example, last use case is to identify cryptic pockets. So not all proteins are druggable. There are proteins that we can't basically design a drug to because they don't have these pockets. When we do molecular dynamics, we model the dynamic of the protein in solution, and basically we can find these transient pockets and design drugs to. And because it's so computationally expensive, what we can do today is use these trajectories from the molecular dynamics and basically train machine learning models to predict these pockets. And with that, I will hand it to Aaron. Thank you.
Thanks, Danny and Matt. It's really cool to see things like, obviously, biofems, but also more traditional approaches like MD be applied in this space. I remember, 15 years ago doing, some of the early MD work, and it's really cool to see that, it's all coming together, right? You run the simulations, you use that data as effectively, synthetic or pre-computed data in some cases to then generate new models for new predictions, and then being able to scale those as well. So we're early on in reInvent. We're, I think, in the second day, obviously, and so there's a lot of sessions that are still ahead. We've put a few of them up on the slide. Some of them today, if you're interested more in HealthOmics, there's a great talk by one of my colleagues, Margaret McDowell, with Natera, on how Natera is scaling, some of their diagnostic testing on HealthOmics. But there's a broad set of things I would highly, highly recommend taking a look at our innovation talk on Thursday by Dan Sheeran, but also just a broad set of chalk talks and workshops, whether you're interested in foundation models, whether you're interested in more general AWS for healthcare and life sciences. There's a bunch of things today. Feel free to scan that QR code. We'll leave that up here so you can use that and get immediately linked to our attendee guide. And with that, I want to say thank you. Obviously, this is there's a lot here in the next 3 days. We very much appreciate all of you being here. We hope you get something really out of it. After I conclude in a second, we will go to the side for any questions you may have. Also, feel free to go grab lunch, get your next session. I recognize we're in the lunch hour. But again, thank you for attending. Very much looking forward to, personally, for all the other great talks and hearing some of the excellent customer stories. And I hope you have a wonderful reInvent.